today's presentation was focused on the clinical perspective when it comes to biosimilars. It started with the clinical trial design because normally we have learned to go from phase 1 to 3 and 4 and then bring it to approval, and now we have to learn that we have to do pharmacokinetics, pharmacodynamics, and then just do one decisive clinical trial that is really necessary for approval. For the approval trial, of course, we need an endpoint. This is normally response rate. Coming from metastatic disease or from early breast cancer. And early breast cancer is certainly the sensitive population because there's just a primary tumor to be measured. So this is a very safe population that if there was a difference, we would notice it between the comparator and the originator product. The response rate has to fall into a predefined range of margins that are defined by the confidence intervals. The confidence intervals can be chosen by the manufacturer, but it must really be in that range and cannot be outside this range that would define a lower or a higher efficacy. We know that the trials, of course, even in early breast cancer, are not all the same. There are trials with a switching process and some without, and the Amgen trial has a predefined switching randomization. So altogether we have 3 populations in this trial, one on the originator product, one on the biosimilar, and one that switched within the trial, and we do not have any signals that there is some difference in safety or immunogenicity between the 3 arms. Last but not least, extrapolation is always a topic because we normally do a trial in one tumor entity, but then transfer the results and the reliability of the results to other approved indications. In breast cancer, this would be gastric cancer, HER2-positive gastric cancer. This is also being approved together with breast cancer. It will be even more interesting with bevacizumab because there the clinical trials will be done in non-small cell lung cancer and then extrapolated to all the approved indications, which is ovarian and breast and colorectal and bladder cancer. So just one trial that confirms the efficacy and the equivalency and then extrapolated to other tumor indications and therefore provide savings for the healthcare systems. Well, for me now, in our current situation, there's absolutely no reason to use an originator anymore because the efficacy and the safety profile are the same, and that's why shouldn't we save this money to use it for our patients in other indications with other diseases, or to save this money for the healthcare system. It is equivalent, and normally we do not waste money. It doesn't make any sense. I think in the future we will be so used to everyday usage of biosimilars that we will have forgotten to talk about it. It will be the standard. Now it is still somehow new, but in several years I think we will not talk about it anymore. For me, the patients nor the physicians are reluctant to use it. I know my patients quite well. The oncological patients are very reasonable. They know that the social system provides medications that they could never ever afford themselves. That's why if there is a chance to save money, they normally accept it quite calmly and knowing that it is needed at other people, other indications, for other reasons.